Hello and welcome to biology. So the first thing we need to look at is the difference between prokaryotic cells and it's not the best ampersand in the world, eukaryotic cells. Let's try get the handwriting a little. Okay, so there's prokaryotic and eukaryotic. So prokaryotic cells, single celled, e.g. bacteria. How about eukaryotic cells? Well, multicelled, e.g. animal and plant cells. And plant cells can be seen as like animal cells, but with more stuff. Okay, so I've just looked and I'm just going to write this here as well. So yeah, I have a feeling there is something a little more to this. Just putting multi-celled here is perhaps not the best way to do this. But then again, all multicellular organisms are eukaryotes. Yeah. All right. So, what's next? Well, what's next is that I'm going to be drawing a picture. And like I've mentioned, okay. I'm going to try to trying to use the paint bucket here. Try and get all right. So, I'm going to actually open up the menu here. I think that might be open now. Hopefully it is. It's not just overlaying it. So what I'd like to do, I think, is to start with some purple. Actually, I have a black line, and I'm going to use the same stroke width because it might be a bit easy to do it like that. In fact, I could actually use this shape here. Yeah, why not try that? So what I'll do is I will first of all create a black outline with a purple fill, and I'll create this shape here. I'm not going to go overboard, I'm just going to make it like that. And the fill should have been put in there, but it hasn't been. There it is. Okay. Now I'm going to take a second rounded rectangle. I'm going to change the fill colour to a light blue. And I don't want to leave much space really. Just going to try and put. Let's go to the paint bucket here. Okay, looks good. So the first thing I'm going to want to point to here, I'm going to go back to, I believe I just go back to my pencil, might be okay. What I might do actually is draw, I'll do all the labelling afterwards. Maybe I won't actually. However, what I can do is I prefer to use red there. I prefer red. So I'm going to try and do some red here. Oh. I wonder what tool I was using before then, if it was like that. Well, that thickness is unacceptable, really. So is that. Alright, let's just do that then. Alright, well that looks a bit better. Let's just make sure. Well, yeah, I think that's, I think that's clear enough. Alright. So this is obviously to show what was known as the cell ultrastructure. And the ultrastructure is kind of a collection of all the different organelles, and organelles are particular features of the cell. This is the first feature I'm going to talk about there, the plasma cell surface membrane. And I'm going to do this in such a way that, yeah, okay, so I'm just going to put, let's just lower this down a little. Well, animal cell, there we go. Okay, so yeah. Okay, so now what I'm going to do, I'm going to, I may as well use as many of these things as I can, really. I'm just going to choose a slightly different colour blue here. And I'm going to put, I don't have to do that. I'm just going to put that in there like that. And I'm going to paint bucket. And yeah, I'll change that there. Do something like this. Okay. And wonder how this will look. Let's have a look. Oh, did not want to do that. Alright. Alright. So I want to put all these, well, little dots. And now I want to have nucleolus here. Good. Yeah, that's pretty cool. Alright. So now, and I kind of figured it's almost in a way easier to do it this way. I'm going to freehand the nuclear envelope. So let's do this. Alright. Yeah, and I suppose in time I'm probably going to learn a lot of these different shortcuts. Use optimization. Alright, so I will actually Alright, so that's the nuclear envelope around it. I think I've actually decided I am going to just draw all the stuff first. Alright, so I've got the nucleus is obviously the this blue area, the dark blue area there. Nucleolus, which is obviously the black thing, black ball there. And the nuclear envelope is the yellow around it. Okay. And obviously, okay, let's do some ribosomes now. Okay, ribosome time. And yeah. Oh, not that. Yeah, it is actually a lot easier just to stick with one colour and just make it feel everything massive. Rather than try to do fill colours at the same time, because it's not really. Alright, so I'm going to just do lots of spots all around here, just to show ribosomes all over in the cytoplasm. Okay, so that's fine. Now, what should I do? Mitochondrion or... yeah, mitochondrion time. So these are slug-like creatures. Yeah, they look quite similar to... well... and I'll make 4 of these. Okay. I'm not leaving too much space for the smooth endoplasmic reticulum and the Golgi apparatus. For some reason, I'm trying to think how. Yeah, with Golgi apparatus, I think. Did I used to? Yeah, did I used to say apparatus? I'm sure I did say apparatus. And Golgi. I want to say Golgi, but I know it's not Golgi. I know it's Golgi. It's just a little one there. Anyway, there's the mitochondrion. And let's have some lysosomes. There you go. This is an animal cell, of course. Plant cells are a bit more complex. And I'll probably draw them next time. Yeah, I'll probably end up drawing these things quite often. I don't think it makes too much difference if there's a border or not. So we're going to do... I'm not going to have too many lysosomes, actually. I'll put 5 because I don't want the same number of lysosomes as mitochondrion. But, yeah. Now, what else am I missing? Not much really. Golgi apparatus and smooth and double ribosomes. Alright. So now doing... I haven't really left that much space. Yeah, these items now are going to be a little bit hard to do, and I have a feeling I'll probably have to draw them again another time. So I'm going to put Golgi apparatus here. I'm going to have to lower this down there. So there's the Golgi apparatus. And then we're going to have the smooth endoplasmic reticulum and then the rough endoplasmic reticulum. Okay. So here we are. Very smooth. Now, rough endoplasmic reticulum. Okay. Now let's label everything. As you can see, I've got the... I'm going to actually... I might try... what fonts? Yeah, okay. Okay. So, I'm going to go in here. Let's try and get this as neat as I can. So there's a rough endoplasmic reticulum. Nucleolus. Ooh, now this one's going to be a bit tricky. You know, nucleus, yep, cool. You know, that ribosome didn't quite make it. Oh, I've been a little bit cavalier. All right. Oh, lysosome. Okay. And obviously, Yeah, ribosome. I'm going to explain all these in the next episode, of course. There's nuclear envelope and Golgi apparatus. Golgi apparatus. Yeah, I want to say apparatus. I don't think apparatus. And of course, this random blue fluid, cytoplasm. Okay. And finally, Mitochondrion. So there we go. That is structure and ultrastructure of an animal cell. So I've explained the differences, prokaryotic and eukaryotic cells. Obviously, work for further learning. Which is interesting in the first episode, but I guess that is the nature of biology. It's always further learning, but then it's probably all subjects. Now we have the cell membrane, the rough and the smooth endoplasmic reticulum. I don't want to pluralize that though. Could do. The nucleus, nucleolus there, inside the nuclear envelope. These ribosomes here, lysosomes, mitochondrion, and all